So I'm going to skip the technical slides, just give a brief introduction to Rob. He is a plant disease epidemiologist at Plant and Food Research. He is involved with both the Bioheritage, Ngā Raka Takitaki and Beyond Myrtle Rust research programs. He undertakes a range of projects that draw on his extensive experience in plant pathology. And within these programs, he is working on understanding the biology of Apicidiae and the interactions of this pathogen with hosts and climate. So I'm looking forward to hearing more about this today, and I'm hoping the universal thumbs up signal will lead to Rob commencing his presentation.
Kia ora tātou. Actually, I can hear you now, Renee. Seems to be sorted, so everything's good. Thank you.
Excellent. I think you should just commence.
Okay, thanks very much, and thanks to everybody out there for listening today. It's a great honour to be asked to give this presentation. I hope it'll have some stimulating information in it and promote lots of discussion. So it's about predicting the future spread of myrtle rust in Aotearoa, New Zealand. Okay, hang on a second, I just have to. Here we go. So myrtle rust's been here for 4.5 years since it was first detected in 2017 on Roll Island and then in Northland. In 2020-21, there's been more awareness of it than at any other time since the initial MPI incursion response ended in 2018. Myrtle rust has been found on more new hosts at more locations in infected areas and over a wider geographic range from the far north to Christchurch. And Rebecca Campbell at Plant and Food in Motueka very kindly updated this surveillance map for me yesterday. So it shows all the myrtle rust records dating right back to the beginning of the incursion. So the MPI doc records and more recently the iNaturalist New Zealand records, which is our main repository now of myrtle rust surveillance information. So the number of observations has ramped up in 2020-21. You can see this reflected in the iNaturalist records. We have to remember here that the awareness of the app has actually also increased, so that's contributing to the number too. But there's lots of evidence that this last year has been a great increase. And it's interesting to look at the seasonality of that increase, or the distribution, because most observations happen from November to May. So November through till May is when there's a lot going on. From July to September, very few, and right now, today, in fact, the middle of September is the annual minimum activity of myrtle rust. So we're in a little bit of a quiet patch just now. So the increased activity may be due to seasonal weather, and particularly some warmer winters that we've had. That's probably been contributing. There's increasing inoculum load resulting from gradual spread of the pathogen over time. So the question is, how will this continue to play out, and how can we predict future impacts? So this talk explores existing epidemiological knowledge of myrtle rust and other rust pathogens to understand mechanisms of spread and how these can identify new approaches to predict the future impact of myrtle rust in Aotearoa, New Zealand. Spores are the only means of spread for rust fungi. Uridinyl pustules produce asexual uridinia spores, which are the main means of spread for myrtle rust. Field monitoring shows sexual teliospores are occasionally found in New Zealand, and Michael Bartlett and Sion has reported on this, but their role in disease spread is certainly unknown and probably negligible. Uridinia spores are adapted to dry, wind-borne dispersal. Most uridinyl pustules form on the lower leaf surface, which protects them from rain. And they're ornamented with hydrophobic spines that enhance adhesion to plant surfaces when deposition occurs. And that nice electron micrograph is by my colleague Paul Sutherland in Plant and Food, and was funded in the NRT program. However, although they are adapted for dry, wind-borne dispersal, they can, of course, be spread by other means. Human activity is an important one, via clothing, equipment, during disposal of infected plant material, and by transportation of infected nursery plants. Bees collect rust spores, including myrtle rust, and you can get cross-inoculation between flowers and hives. The epidemiological significance of this is not understood. Rain is possibly the least important means of spread dispersal for rusts. Prolonged or heavy rain may actually decrease spread by damaging Eodynia and washing spores to the ground. So how long do spores survive in the natural environment? The key question regarding risk of Myrtle Rust spread is how long do the spores survive? In the laboratory, they can be stored under artificial conditions, such as -80 degrees, and remain viable for months or even years. However, the natural environment is very different. Nighttime wet periods occur because of diurnal fluctuations in temperature and humidity, and these promote spore germination and infection. Rainfall also contributes to wet periods. So once the infection process starts, germinating spores will desiccate and perish if drying occurs before the infection is complete. And that nice micrograph there shows a couple of Eodynia spores on a Pahutakawa leaf infecting, producing the germ tubes and the aprosorium, which is the infection structure, and penetrating the leaf from there. Unpublished field inoculation experiment that I did in 2018 showed most spores infect during the night after they are deposited. If it's too cold or the wet period is too short, a few will survive to infect on a second night. Spores that land on a non-host surface will germinate when humidity is high, then die. So Eodynia spores are probably short-lived under natural conditions. So looking at long-range wind-borne spread, so Myrtle Rust is one of several rust fungi that are believed to have travelled by wind from Australia to New Zealand. Evidence supports this for rusts of Antirrhinum, blackberry, oxalis and poplar, and stem rust and stripe rust of wheat. And there's a number of good publications on that phenomenon. In 2017, NIWA modelled trans-Tasman spread of Myrtle Rust, and they found that Australia's east coast was considered the main source of risk, rather than Royal Island or New Caledonia. And of course, at that time, that was the very big interesting question, was where had it come from? The most exposed areas were considered to be Northland, Taranaki, Tasman and Bay of Plenty. And the second most exposed areas were Auckland, Waikato, Wellington and West Coast. And the early Myrtle Rust distribution more or less matched this analysis. So it seems reasonably sure that Myrtle Rust got here by that means. But how far do the spores usually travel? And as many published studies show that spore concentration decreases exponentially with distance from an inoculum source. Dry airborne spore types travel further than wet rain-splashed spore types, but deposition gradients from spore sources are always very steep. It appears that the number that escape into the atmosphere and travel long distances is relatively very small. So whether Myrtle Rust arrives in a new area by human activities, insects or animals, or by long-range wind-borne spread, it subsequently spreads locally over a short range and becomes epidemic on susceptible myrtle hosts. Observations show that when first detected in a new area, it can be locally very intense, but often limited to just a few shoots. Published research work gives us an insight into the mechanisms that operate to cause this initially restricted spread. And there was a nice study done in 2017 in the USA on the spread of wheat stripe rust over short distances. So spread of rust as new lesions derived from individual mother pustules was measured. And most infections occurred over a remarkably small spatial scale, and the graph shows it quite nicely. This is the distance from a mother lesion, and this was done over a 3-year period with an area where it was isolated from rust infection normally. And you can see that the vast majority of spores travel a very small distance, about less than ½ a metre, and causing infections in that very small area. It explains why rust epidemics, including Myrtle Rust, develop from point foci rather than from evenly distributed infections. Looking at disease spread over time and space, I was involved in a trial on peppermint rust in Canterbury in 2019. The farm was growing peppermint organically for essential oil production. We tested benign fungicide chemicals for rust control. Unfortunately, they didn't really work, but by chance, the disease data provided an interesting opportunity to examine the spread of rust from the focus of infection. The trial area was long and narrow, 80 metres long and 4 metres wide, so there were 20 plots along an 80 metre stretch. This is a 3D graph rather than a diagram of the plot, so that the axis going out this way is not the plots, but it's time from the 4th of February through to the 18th of March, which is when assessments were done. So rust infected the trial at one end and spread gradually along the plots over time. Fortnightly disease assessments were made for 1.5 months from the first sign of rust until harvest. The velocity of horizontal spread in metres per week is a parameter that describes the characteristics of an epidemic in space and time. It can be measured as the time it takes for a certain disease level, or an isopath, as we can call it, to travel different distances from a point source of infection. For example, the 40% rust severity isopath in this diagram. So it took from the 16th of February to the 18th of March, 30 days, for the 40% isopath to move from plot 20 to plot 13, a distance of 28 metres. So in 30 days, it's travelled 28 metres. That's a velocity of spread of 6.5 metres per week. And that's an interesting number to ponder, because depending on your scale of reference, it's either very fast, like it was moving down those plots pretty quickly. It wasn't moving quickly enough to even it out so we could do the fungicide trial well enough, but it was moving quite quickly down those plots. On the other hand, on a landscape scale, 6.5 metres in a week is not very fast at all. And there's another interesting way to look at the velocity of spread as well, and that's in relation to the infection cycle time, or latent period. So the mean air temperature during that period in that area was 17 degrees over the assessment period, and the infection cycle time, or the latent period, we know would have been 14 days, and that was from some earlier work that I'd done in the early 80s, actually, at the beginning of my career on mint rust and peppermint. So that meant that there was time in 30 days for 2.1 infection cycles, with a velocity of spread of 13.1 metres per infection cycle. And that's an interesting number, because in the time it takes for the fungus to infect and produce new spores, it can move 13.1 metres in that situation, which again could be slow or fast depending on your scale of reference. There was a very elegant study of that very same thing done on striped rust of wheat in the USA by Christina Cowger and her colleagues in 2005 on the spread of striped rust in pure stands of a susceptible cultivar. They were inoculated in the middle of the field to create a focus of infection. The disease severity was measured in different directions over time, and the velocity of spread calculated from the isopaths. It was faster spread downwind than upwind, which is shown quite clearly in this graph. The downwind direction, the spread was going reasonably quickly, upwind very much less so. And the velocity of spread increased over time. At the epidemic midpoint, it was about 3 to 7 metres per week, which interestingly is pretty similar to what we found for the mint rust. And that the fact that it was increasing over time was quite an important finding, because epidemiologists had been arguing for some time about the rate of velocity of spread, whether it was constant or increasing. Now, they also did the same thing in mixed susceptibility trials, where there was a susceptible cultivar mixed with an immune cultivar. And in the immune one, of course, or in the mixture rather, the spread was very much slower. Velocity of spread increased slightly over time. At the epidemic midpoint, it was about 1 to 3 metres per week. So myrtle hosts, like the wheat cultivar mixture, are usually not pure stands. And although the velocity of spread over space and time would be different for myrtle rust in the natural environment, the mechanisms of dispersal and spread are the same and could be modelled given the right data. So there's an interesting paradox that spores of myrtle rust and other rusts can travel across the Tasman Sea from Australia to New Zealand. Yet, during an epidemic, almost all spores are deposited over a very short distances, and the spread is gradual from point sources of infection. To resolve this paradox, we need to realise that the epidemic spread from a focus of initial infection is not just about the travel distance of spores. Spread of damaging disease across a landscape depends less on how far spores can travel and more on how fast the pathogen can multiply, which is something very important to remember, which is obviously points to the reason why latent period is such an important parameter in rust epidemiology. So in terms of disease spread and detection, long distance windborne transport may get myrtle rust to a new area, but it won't be as a cloud of spores hitting a population of susceptible hosts and causing widespread damaging disease. There will be as a few widely dispersed spores that form infection foci and require repeated cycles of infection, latency and new spore production before the incursion is even detected. The problem that hinders our understanding about mechanisms of spread is our poor ability to detect small amounts of disease, and better surveillance methods are really needed for early detection, whether it's in a plant nursery or in an area of rust-free native bush. Now I want to turn now to host factors affecting myrtle rust spread, because these are as important as anything. And the first one, of course, is genetic susceptibility or resistance of the host. It's how a plant reacts to infection by Osteococcinia acidii as a result of its genetic makeup. And it's estimated from the phenotypic reaction to infection under standardized inoculum temperature and humidity conditions. The earliest one of these studies I know of was the Brazilian one by Ongenic et al. And these are the categories that they put hosts into, ranging from immune, highly or hypersensitive reaction through to highly susceptible. And this has been done for some New Zealand native species through seedling screening in Brisbane, Australia. And Grant Smith and company have published a paper on that for some of our species. But then the second important host factor in terms of susceptibility or resistance is ontogenic resistance. And it occurs in the Myrtaceae to myrtle rust. So shoots change from susceptible to resistant after they emerge and are completely resistant to infection at about the time they reach full size. It limits infection and myrtle rust epidemics to periods when seasonal growth flushes occur, and that has a huge impact on how it spreads and when it spreads. So looking further at host issues, density and spatial distribution of the host is a really important one, obviously, especially for something that's occurring in the natural environment. Basically, higher density equals faster disease spread, as was alluded to in the fewer stands with striped rust versus the mixtures earlier. Closer host spacing equals decreased dispersal distance, more spores deposited, and fewer spores lost to non-host plants into the ground, into the atmosphere. So host density is a critical parameter for modelling velocity of spread. And then the other one is spatial distribution. Now, Myrtaceae occur naturally with clumped spatial distributions, making modelling the effects of host density on disease spread quite difficult. But empirical spatial indices could be used with field experiments, measuring disease over time and space to develop landscape scale prediction of myrtle rust spread. This would help with predicting myrtle rust impacts and also with surveillance planning. Epidemiology and ecology would need to focus together on common objectives to achieve this, and this hasn't been done anywhere for myrtle rust to date. And it's something, it's a new area of thought that I think we epidemiologists and the ecologists need to give some serious thought to. So just looking at the likely future distribution of myrtle rust in Aotearoa, and this is my best bets, I think, really. In 5 to 10 years' time, the distribution may look like the red and pink areas in this map. And the map, of course, was the one we did back in 2017 or 2018 on the climatic risk. And this was the maximum summer risk depiction of climatic risk. And it shows that myrtle rust could well be from the far north all the way down the west coast, all the way down the west coast of the South Island, to the northern South Island, Canterbury and other coastal areas in South Canterbury. I think this is probably where it will end up. But although it will be more widely distributed than at present, it may not be very damaging in the more southern areas because of a shorter season for epidemic development. The other point about climate, of course, is the climate change and myrtle rust spread in the future. The climate warming, we know this will increase myrtle rust risk. In a recent report to MFE that Rebecca Campbell wrote, Plant and Food Research in NIWA analysed how much myrtle rust could increase under a range of future temperature increase scenarios from 1 to 5 degrees. And the increasing temperature will increase the number of days suitable for infection and increase the rate at which infection cycles occur, which will cause more rapid spread further south into higher altitudes, and increase inoculum load and decrease severity in currently marginal areas. So looking at likely scenarios for future spread into new areas, and I think this reflects what we've seen before when we look back in retrospect. So development over multiple seasons, where a season runs from July to June. First season, undetected arrival occurs, most likely in late summer when the disease level in the source area is highest, and most likely to infect highly susceptible host species. Small-scale local spread establishing a focus of infection, and then first detection at some point. The timing depends on how long before winter it arrived and whether there is active surveillance occurring. In season 2, the initial focus expands, rust presence in the area becomes obvious. This third season, increasing local spread and severity, inoculum load increases. In the fourth season, detected on less susceptible species, and the inoculum load keeps increasing. And I think this is where we're at with our current outbreak of Myrtle Rust, that has now been seen on the less susceptible species, and then that will, I think, continue into the future. And so in season 5, we've got severe impact on highly susceptible species and the infected area expanding and infecting less susceptible species as well. So just to conclude then, spatio-temporal disease models hold the promise of predicting Myrtle Rust spread at the landscape level. And this is a new area which hasn't been looked at, and I think is really going to be needed for our in-depth understanding. They need to be coupled with spatial ecology modelling to represent the spatial distribution of Myrtaceae hosts. While it was initially said that Myrtle Rust is quite different from other rusts, in fact the perceived differences are probably due to differences between monocultured crops and natural plant communities. Because the biophysical mechanisms are consistent, learnings from rusts on other hosts have a great potential to help us understand and deal more effectively with Myrtle Rust. So thank you for listening. And I'd just like to acknowledge these funders and contributors.
Thank you very much, Rob. Seems that we got there in the end despite some difficulties. So can you hear me okay now?
I can hear you fine now. Yes, thanks,
okay.
Renee.
Excellent. So for those that might not be very familiar with the GoToWebinar platform, you can put your questions in. Now you'll see in your little control panel on the side there is a question box. It's really small. You can pop it out by clicking the wee box in the corner. Enter a question. I'll be checking those questions, sharing them with the audience and giving them to Rob. So we have got one question in so far, which is... Could
I just ask, Renee, are we still sharing the screen?
We are. Yep.
Do we want to keep sharing it, or do we want to see me?
Oh, we can see you as well.
You can see me?
I think the screen's fine just as it is. It's all good.
Cool. Okay, that's good.
And the question that I have here is, given the short life of spores, does the rust overwinter as slow growing infection within the host? So a good chance perhaps to talk a little bit about what is going on in winter with this pathogen.
Yeah, that's right on the mark. That's exactly how it overwinters, as latent infections. And we've done some fairly thorough work on that now for the New Zealand environment, and actually in conjunction with our colleagues in Brisbane and QDAF, we were able to show that at temperatures below, it depends on the species, below about 10 to 12 degrees, the development of Myrtle Rust is so slow that it effectively stays latent.
Would a hard frost kill it?
Yeah, well, I think that it'll probably survive any temperatures that don't kill the host. That's my guess. And we know that some of the hosts are very frost sensitive. Aotearoa, we've done trial work. We've done observations on Aotearoa that we've planted in different parts of the country. And for example, in Motueka and also at Rotorua, frosts kill it in the winter, unfortunately, makes our studies more difficult, whereas the Lophomatha species seem to be barely affected by frost.
Okay, so some more questions have started coming in now. Ricky, some Tricky ones here. You know, the fungicide question, of course, is important to a lot of people and unfortunately not well resolved yet, but I'll just put this one to you. I have numerous eugenia around my garden, large hedges about 5 meters, and some smaller trees. I've tried commercial products with very little success. Do you have any suggestions?
Depends which commercial products you've tried. I think I mean, we do have a pretty clear idea of which fungicides work well against myrtle rust because of work that's been done here by Scion and also work that's been done overseas in Australia and in Brazil. There's one commercial product available from home garden shops that I know of, which I'm not 100% sure if it's still available. It's a thing called Yates Fungus Fighter, and it's a triazole fungicide, and it's extremely effective. Now, if you're using things like copper or some of the older fungicides like Captan or Mancozeb and those ones, they will have some efficacy, but they won't be particularly effective.
I do have an add-on to that. They have tried Maverick.
Oh, right. Okay. Well, Maverick's an insecticide. Maverick's an insecticide, so that will do nothing against myrtle rust. Yeah.
Probably not. All right. Next question. Oh, okay. Bees. Yes, we've got a couple of people chatting here about bees. I read somewhere that bees preferentially harvest from myrtle rust infected plants. Can you tell us a bit more about the risk of transmission by bees? And how significant are these anyway, given that myrtle rust is spread by wind as well?
Yeah, I'm not the expert on bees. I believe there was a study done in Australia which showed that that they could find viable myrtle rust spores inside beehives. So it clearly happens. And when Maverick is flowering, of course, there's bees, can be bees everywhere, and it's very spectacular. So there must be a lot of opportunity for bees to spread spores from one flower to another, and we know it can infect flowers and fruit. So that would apply to all the myrtles because they all have fairly similar sorts of flowers. But as the question alludes to, the importance of that in relation to the natural windborne spread is unknown. It would only be while there are bees active in the area of myrtle plants, whereas the windborne or airborne spread is going on constantly all the time. So I don't personally think that bee transmission is probably a major source of spread.
Okay, next question. Would you recommend removing the infected part of the plant if myrtle rust is detected?
Yes, I would, and removing it carefully. In fact, I don't know who's asked the question, but if they've looked at the MPI website, there's a lot of information in very detail about how removal should be done. I think it starts with using hairspray.
Yes, I mean, perhaps there's going to be some opportunity soon to bring that information a little bit up to date, because it does get very, very difficult if you've got extensive foliage to effectively apply a hairspray or perhaps even a fungicide. But yes, the message really is in urban gardens, at least I think, becoming clearer that these garden plants are sources of inoculum and they probably should be removed. So yeah, similar question really, when the infection is latent, is it still localized? So would removing the visibly infected area actively treat the infection? I guess that's largely the same.
Yes, just on that one, wherever there's active infection, there's latent infection. Probably roughly around about half the infection on the plant will be latent and half will be visible at any one time. So yeah, removing it, visible infection will remove a lot of the rust, but there will be some that will be in parts of the plant which are symptomless at the time. So just bear that in mind. Sometimes it might be better to remove a whole plant, especially if they're some of the exotics which are of less value to us in New Zealand.
Okay, most epidemiological studies have been on host-specific rusts. How does the wider host range affect myrtle rust epidemics?
Yeah, is the host range wider? It's certainly you know, it doesn't affect anything other than myrtles, and there's only about roughly 10 or 15% of myrtles which are susceptible, it seems. So Yeah, what I think the way we need to view that one is that the hosts of Myrtle Rust, which we don't fully understand yet, but the ones that are susceptible, susceptibility is hugely variable. So we've got this variation that's almost a complete continuum from even within species and between species of susceptibility. So that's challenging, and the biggest challenge of that is knowing what is susceptible and what isn't. And we need to do a lot more work on that.
For sure, right. We're really getting a lot of questions coming through now, Rob. So hopefully you're okay to stick with us for a few more minutes.
Oh, sure, yeah.
Okay, we'll see if we can get there. And of course, lots of thank yous. We have someone from Sydney letting us know we see infection all through winter, so the rate of spread, I guess, is therefore higher.
Yeah, yep. We know that where it's warmer in the winter, it can keep cycling. Yep.
Evidence of feeding birds spreading Myrtle Rust spores. Has anyone studied that? Do you know? I'm not aware of any of those studies myself.
Not aware of that one, no.
Okay, is it worth, I'll just share this one. Local suburban groups encouraging gardeners to remove lily-pilly monkey apple to lessen the stepping stones between bush areas to help protect those areas.
Yeah, I'm all for that. Yeah. I think it's a very important thing. The problem, well, the advantage of those species is that they're wonderful hedge plants and they're very popular because of that. You can prune them, or you have to prune them often. You can prune them like mad and they just bounce back again, and they don't have big dead gaps in them and things like that, like a lot of other species. And there really isn't anything that's quite as good as the lily-pillies and Eugenias and those. But I think that we should be discouraging their use in New Zealand.
Yeah, we're certainly, as a research program, looking towards more of that. Now, Ongenic resistance. I was under the impression some highly susceptible hosts in Australia were susceptible to Myrtle Rust, even in mature, fully expanded foliage. Is that something you're familiar with?
Well, I'm not familiar with that, and we've done quite detailed work on this. One thing that can happen is that rust pustules can keep appearing on fully expanded leaves after they're fully expanded. So it doesn't look like it's only the young ones that have been infected, but in actual fact, what may have happened is that they became infected when they were small, still expanding, and then the latent period is such that they didn't appear to be infected until sometime after they'd finished expanding. And then on the other hand, you do see rust pustules on old leaves, because once they appear, they hang on. And if it's not a high density of infection, just a few pustules on the leaf, they can actually survive as long as the leaf survives, which could be months. So I think that's why it can be confusing, I think, as to exactly what much is susceptible.
Yep, it's certainly a complication. Right now, from your predictions, Rob, it looks like Myrtle Rust is going to essentially spread across New Zealand within 10 years. That doesn't give us much time for breeding programs. Well, yes. How do we manage Myrtle Rust? Once this is the case, will some of our very susceptible species be near extinct in that time? Would you like to make a call on that?
Yeah, no, well, I think they will. It's not what we want to hear, but I think local extinction may well happen, and people like Ryan Sutherland, who was with Cyan and now with Doc, has been doing perhaps more observation in the natural estate than anybody, and yeah, she's suggesting that too. So I think we are in that sort of situation. It'll be worse further north where the temperatures are warmer, and maybe further south it won't be so bad. And you can do well to read James McCarthy's Refugia paper, which is about whether there are areas in New Zealand where Myrtle Rust might exist, but it doesn't damage the hosts.
Okay, excellent. It looks like we've just got a couple more now. So on that note, someone's asking what we do know about the resistance in native species, what species are most at risk. Perhaps you'd like to rattle them off for us.
Okay, yeah, the list is incomplete, but the two Lophomertus, Octodata and Bullata, are probably the most susceptible we have. Metrosideros excelsa Aotearoa is very susceptible. We know that some of the climbing ratas are somewhat susceptible. I don't know that there's much evidence about northern and southern rata. I think that they may be more resistant.
Well, we're starting to see infections on rata, I believe.
Oh, well, it's certainly the climbing rata, yeah, for sure. Swamp Maori Tawaki is very susceptible, and we're quite fearful about that. That causes, it can kill off leaves and flowers and everything, and it did last summer. Bartlett rata, which is our very, very rare species, done some inoculation trials on that with Auckland Botanic Gardens, and that is fairly susceptible. I would say about the same susceptibility as some of the climbing ratas. Where does that leave us? I think that's probably the main list, yeah.
And looking forward now then a little bit, things we can do. Any ideas about how we might go about detecting the small early infections?
Yeah, I wasn't going to hazard a guess on that. There's people with different skill sets that are needed to do that. Possibly some kind of molecular detection techniques, but certainly a better understanding of spread and the processes involved, I think, can help us target surveillance better, and just frequency and awareness. I think, and the other big one in some of the areas that Myrtle Rust hasn't got to is to upskill people in recognising symptoms and being aware and looking for it.
So yeah, perhaps that's partly an answer to the next question. Do you have an idea of what a good first step would be to build knowledge on how infection spreads when you consider multi-host dynamics and inoculum load? Do you know of examples of this for other pathogens? So perhaps she's asking about more big picture studies for other diseases that we could draw knowledge from. I think you were saying it's not really been done much, if at all.
Oh, well, not for Myrtle Rust, that's what I meant. It's been done extensively for cereal rusts, probably the most intensively studied. Soybean rust would be another one. So they're in crop situations, but as I was trying to say in the talk, it doesn't necessarily matter because the physical processes are the same for rusts. I can conceive some field-based experiments with planted myrtles, where we could look at different plant densities and degrees of clumping over smallish spatial scales, maybe 50, 100 meters, something like that, and we could actually gather some data which could tell us exactly what rates of spread look like, and I think that would be a good starting point.
And on that note as well, should Myrtle Rust be reported to someone? If so, whom to? Well, right now, iNaturalist is it. Really?
Yeah, iNaturalist is certainly where our records are going. If it's in a new area or on a new host species, MPI wants to know about it still. So there's no harm in talking to MPI, calling the hotline if you're in any doubt about that.
Okay. Question again. This goes a little bit back to removing problem plants. I think when it comes to exotic Myrtaceae, it's fairly easy to answer that question. Natives gets a bit trickier. In Wellington region, we have a huge amount of Pahudakawa which are not native. Would it be beneficial to our local native Myrtaceae if we got rid of them?
Yes, indeed. Some pretty hard judgment calls are required here, and there's many very difficult questions. I personally, would favour retaining natives where possible, even if they're not, even if they've been naturalised artificially in an area. But that's a judgment call, and that's a community decision. I think, I think it just requires a lot of discussion, and we haven't really started that discussion in earnest yet, and I think it needs to happen.
Okay, I'm just going to do two more people, and then we are going to need to wrap this up. Has there been any work using EDNA testing to increase detection chances near existing known infections? Do you know of any? I don't know of any.
I don't know of any specifically. I don't see why it wouldn't be possible. But one question about that sort of detection is how do you interpret the results? Whether it would detect live or dead spores or what? There's lots of questions.
And lastly, how would I protect our stand of 4 Maori Tawaka, and how would I go about monitoring them?
How big are they? If they're big trees, then that's a bit of a challenge. We don't tend to see myrtle rust in the tops of big trees, mainly because we can't see it up there, but maybe also because the canopy expansion rates are small in the sense that there's a high standing mass of old leaves and not a lot of new growth. Maybe the host density is effectively not high enough for epidemics to develop in the tops of trees. That's just a thought, but drone surveillance would be one thing to try. A bit of that has been tried, but it's very difficult to see, certainly difficult to see pustules. However, you could probably see the characteristic dieback patterns of myrtle rust attack. So that's something which could be looked into more in the future.
Excellent. Well, we're going to wrap it up there. That's we're going to call it done for today. Very extensive question session there, and thank you so much, Rob. Rob is very generous in giving his time for activities like this, for workshops and media engagement, and there's a number of us down here in New Zealand that very much appreciate that. A video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days, and will also be emailed to everybody who registered for this session. Now, the next webinar is going to be a little later in the month again. It's going to be Wednesday, October 20th, to suit the availability of my speaker. We will be hearing from an offshore expert again, this time Alistair McTaggart, a plant pathologist and mycologist at the University of Queensland. Alistair also works within the Beyond Myrtle Rust program. He has been undertaking research into Apisidae genetics and reproduction. His talk asks, can different strains of Apisidae outcross on a universal host? This is of interest because that could lead to the sharing of genetic features and the arising of even more trouble for Myrtaceae. We shall see what he has to say about that. Look out for the invite in another month or so. Thanks again for coming along, everybody. It's been a fantastic crowd today. Ka kite, and see you later.
Thanks very much, Renee.